Hello, my name is Thomas Wynn, and I will be covering the Meselson and Stahl DNA replication experiment. So in 1953, the question lingered, how does DNA replicate? There were three theories. The first, conservative replication, where a DNA molecule would get copied and make an entirely new second DNA molecule. The second was dispersive replication, where the DNA molecule would be cut at various points, each getting copied and reattached to produce two DNA molecules, each with some new and some old DNA. And the third was semi-conservative, where the two strands would separate and each would serve as a template to create another strand, producing two new DNA molecules. But which one was it? Many scientists were skeptical that two DNA strands could not separate due to the strength of the DNA molecule. So two scientists, Matthew Meselson and Franklin Stahl, designed an experiment to test which of these models DNA replication followed. They grew E. coli cells in the presence of a heavy isotope of nitrogen N15, which is found in DNA that would make the DNA heavy. They then used centrifugation, which would separate things according to their weight. Initially, all of the DNA in the cell was heavy and at the bottom of the tube since it was grown in the heavy nitrogen. They then transferred these cells to grow in the presence of light nitrogen N14. This way, all of the DNA made in later cell divisions would be lighter. After one cell division, the DNA was half as heavy, meaning that half the DNA molecule contained heavy nitrogen and the other half light. This eliminates the conservative model, which would predict that one molecule would be all light and the other all heavy. After two cell divisions, the DNA molecule was now either half heavy, half light, or all light. This shows that the dispersive model was incorrect, which would predict that the DNA molecule would contain a mixture of heavy and light DNA even after two growth and centrifugations in light nitrogen. Instead, it agrees with the semi-conservative DNA replication model, where every cell gets one old DNA strand and one new strand. Meselson and Stahl revolutionized genetics, which opened the field for genetic testing, forensics, and paternity tests. This was done by identifying the fundamental process by which our DNA gets passed on from cell to cell. And this is why this would become known as the most beautiful experiment in all of biology. Thanks for listening.